to understand better the role of the B-cell receptor, or the mechanism rather, of how the B-cell receptor delivers its signal in CLL cells, we have a structural biology type of approach. So there's plenty of approaches that are extremely valid and do contribute in understanding the signal by the B-cell receptor in B cells, but we chose to look at the three-dimensional structure of the B-cell receptors themselves. So by looking at that, we really see what the molecule looks like. We really get a snapshot of what the molecule looks like. We can understand from the analysis of the structure what which amino acids in the polypeptide chain, that is the protein that is the B-cell receptor, can interact with antigen. And actually our study showed us something, and I can admit it's just serendipitous as an initial observation. B-cell receptor molecules derived from CLL cases have the unique ability of interacting with each other. So rather than having a single antigen, exogenous antigen, may be a virus, another protein from our body, or something else, they do interact with each other. So they act both as the receptor and the antigen, which is a new concept in the field of B-cell immunology, if you wish. So understanding this at the structural level was really important for us, because we really began to understand that we needed to characterize many different B-cell receptors and from different patients and see how they perform this interaction. So that's what we were doing basically right now, determining structures and analyzing this behavior of self-association. We started by observing behaviors that are kind of shared between different subgroups of patients, or if you prefer, B-cell receptors derived from subgroups of patients. And now our new studies are actually showing that this can not be just a general characteristic of B-cell receptors, but each one has its own peculiarities. So on one hand, this is disappointing, because it would be nice to have a common shared mechanism and maybe be able to find just a single antagonist that will work for all patients. On the other hand, this will allow to take us into consideration the individual characteristics of each patient and maybe develop specific compounds that antagonize the association and the signal by the B-cell receptor, which eventually is our final goal.